अटेंड क्यों नहीं है? Hello everyone. I just want to make sure that you can hear me. Can you please raise your hands if you can hear me? Oh, perfect. Thank you. Okay, so we will start in few minutes. Okay, let's start. Hello everyone. My name is Dr. Shweta Pahujani and I am a senior product manager here at BioVision, and I manages BioVision ELISA kit portfolio. Today, we are going to talk about our BioVision ELISA kit portfolio. So the content that I'm going to talk about is, so first I'm going to talk about the overview of ELISA principles. Then I will talk about the different types of ELISA formats available in the market. After that, I will talk about what are the characteristics of an ideal ELISA kit. Then I will give some tips for choosing an appropriate ELISA kit for the users depending upon their samples. Then I will cover some popular kits and some hot topics, some hot targets of our BioVision ELISA kit portfolio in detail. At last, I will give some experimental tips for the users if they have some, if they have some difficulty to obtain results using our ELISA kits. And then at last, we will be having Q&A session. Okay, so let's start. So enzyme-linked immunoabsorbent assay is commonly called as ELISA, is a common immunoassay in which antibodies, peptides, proteins, and small molecules can be detected and quantified using a multi-well plate. The common principle that included in the ELISA kit is the immobilization of an analyte or protein or a small molecule that we want to analyze on the immobilization on a solid phase, and then separation of a bound and free reagents by washing steps, and the readout of signal from calorimetric, fluorescent, or luminescent signals. So the colored end product that generates signals correlates to the amount of the analyte present in the sample. So commonly, there are 4 different kinds of ELISA that are available in the market. So they are classified into direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Out of all 4 these, the main 2 types that can be commonly used by the researchers or for diagnostics or for basic research is the sandwich ELISA or competitive ELISA. So I'm going to talk about in brief about each one of these ELISA kit format. So starting with the direct ELISA kit. So in direct ELISA kit, the antigens are immobilized on the surface of the microwell plate. And then the enzyme conjugated primary antibody are used to detect or quantify the antigen concentration. So as you can see here, the antigen or analyte that we want to quantify is immobilized on the surface of these microwell or 96 well plate, and the primary antibody is labeled that is used to detect or quantify the antigen concentration. So some of the advantages and disadvantages of this ELISA format are, so it's a very quick procedure. It doesn't require too many incubation steps. It's a direct, only one antibody is utilized. So it's a very quick procedure. And as no secondary antibody is used in this assay, so there is no contamination or no issue of cross-reactivity of secondary antibody. However, it has several disadvantages. As the primary antibody in this ELISA is labeled, so that greatly reduces the immunoreactivity of the primary antibody. The primary antibodies are labeled. So the labeling of primary antibody is a very time-consuming process as well as very expensive. And as no secondary antibody is involved, so then it gives a minimal signal amplification. The signal is not that strong, and not many antibodies are available. So limited availability of antibodies, so it can be expensive also. And not every antibody is suitable for labeling. So that means that there are not many antibodies available for the target. So that's it has a very limited choice for the detection of the target. So the next format that has some advantages over direct ELISA kit format is the indirect ELISA kit. So in indirect ELISA kit, the primary antibodies are not labeled, but detected instead with enzyme conjugated secondary antibodies that recognizes the primary antibodies. So in this, the analyte or the protein or small molecule of our interest is immobilized into the microwell plate. The primary antibody is not labeled, but it's bound. It's added to the well, which bounds to the protein of our interest or the antigen of our interest, and then a label secondary antibody is added that is bind to the primary antibody, and then which gives signal. It's mostly this biotin conjugated, which gives signal in the presence of streptavidin conjugated and with the TMB substrate. Few advantages of indirect ELISA kits are it gives more flexibility and versatility. As many antibodies are available, secondary antibodies are capable of signal amplification. So many available secondary antibodies can be used for different assays. Unlabeled primary antibodies retain maximum immunoreactivity. It gives higher sensitivity and signal amplification. Some of the disadvantages of this format are it shows cross-reactivity with the secondary antibody, which gives some non-specific signal or high background. It requires an extra incubation step in the procedure that can enhance the overall experimental time. So moving forward, another kind of for ELISA format that is available in the market and which is most commonly used is the sandwich ELISA. So why it is called sandwich ELISA? Because in this ELISA format, the antigen to be measured is bound between the layer of capture antibody and a layer of detection antibody. The two antibodies must be very critically chosen to prevent cross-reactivity or competition in binding sites. So as you can see that there is one antibody that is specific for the antigen is bound to the microwell, and then the antigen is added to the microwell plate. There is another antibody that is added into the well is a capture antibody, which also binds to the antigen of our interest, and then secondary antibody is conjugated secondary antibody is added, which gives the signal that can be our readout. So some few advantages of this ELISA format are it shows high specificity and sensitivity as two antibodies are used to bind the antigen. So it's very specific and it's highly sensitive. It is suitable for many complex samples. It gives high flexibility as it gives more choice of antibodies. Few disadvantages of this ELISA formats are higher cost and development time due to the several incubation steps involved in the process, and different types of antibodies required for the development of the assay. Longer processing time with multiple incubation and washing steps due to the use of different antibodies at each steps. It requires longer processing and incubation time. So the overall experimental time is longer than the traditional direct or indirect ELISAs. However, this is due to the high specificity and sensitivity. It is most commonly used format by the researchers. And the last format that is also very commonly used by the researchers is the competitive ELISA format. So in the competitive ELISA format, the antigen of interest from the sample and purified immobilized antigen compete for binding to the capture antibody. So a reference antigen that is similar to the antigen of our interest is bound to the microvalve plate, which compete with the antigen with the antigen of our interest, or the protein of our interest, and this antigen compete with the reference antigen to bind to the capture antibody. A decrease in signal when compared to assay wells with purified antigen alone indicates the presence of antigen in the sample. So the more the signal, the less the antigen present in the well. So it's opposite. If more concentration of antigen is present in the well, the OD values will be lower, or the signal will be lower. So few advantages of these ELISAs over other ELISA are, it measures the concentration of an antigen or antibody in a sample by observing interference in an unexpected signal output, and most often used when the analyte is small and cannot be bound by two different antibodies. So it is mainly used for the small molecules, or if the analyte is very small. And few disadvantages of this system is it's less intuitive in calculation, and it shows low overall sensitivity and specificity as compared to the sandwich ELISA. So these are the 4 different types of ELISAs that are commonly, ELISA format that are commonly available in the market, and out of which the sandwich and competitive ELISAs are the most commonly used by the life sciences researcher for the analysis of different molecules, proteins, or analytes. So now the question is, out of these 4, or okay, let's focus on these 2 different ELISA kit formats, sandwich and competitive. How can we determine that which type of ELISA kit is most appropriate for our samples? So what should be the ideal characteristics of an ideal ELISA kit? The characteristics of an ideal ELISA kit. What makes an ELISA kit ideal for the assay? So some of the characteristics are, so ELISA kits should be versatile. So it should have a broad range of sample types such as serum, plasma, tissue, cell culture, supernatant, urine, and many other biological fluids. So an ideal ELISA kit should be compatible with different kinds of samples. Then the second most important parameter in selecting an ideal ELISA kit is a range and sensitivity. So So range, the another point to check is the range and sensitivity of the ELISA kits. So it is important to make sure that they will fit with the target abundance in the sample. So several sensitivity levels may exist for the same targets. High sensitivity range may not be necessarily fit for everyone's need. So it's very important to determine the range and the sensitivity of the ELISAs. So the range in which whether our sample, what is the range of the ELISA kit and whether it is compatible with the concentration of our sample. Then some of the important other characteristics that makes an ELISA kit ideal is the quality controls, which includes spiking results, so spiking results, types of sample tested, linearity assessment, and reproducibility, which includes inter and intra-assay intra-assay percentages. So as a range of ELISA brands is available in the market, it is important to ensure ELISA test quality by checking the quality control performed by the supplier. So these are the four parameters that needs to be ensured by the supplier before purchasing the ELISA kit. And the last is the stability. So it is very important that the ELISA kit should have a longer stability. So any kit less stable, which has stability less than 6 months, is not an optimal ELISA kit to use for. Okay, so again, moving forward, I would like to talk about some of the tips for choosing an appropriate ELISA kit. So first, it is very important to determine the sample species. So sample species, we should know the species of the sample, or whether the kit is compatible. The species of our kit of our sample is compatible with the ELISA kit, or the ELISA kit can detect the species of our sample. Some of the very commonly used ELISA kits, used ELISA kits available in the market are for human, mouse, or rat species mainly. However, there are ELISA kits available for rabbit or goat or monkey, or for different other species. Then it is very important to understand that what kind of analyte needs to be detected. So as I already mentioned, that different kinds of formats of ELISA kits are available in the market. So we need to understand that which format is suitable for which kind of samples. So sandwich ELISA kit is generally suitable for detecting a large proteins with multiple epitopes, such as cytokines. However, a competitive ELISA kit is appropriate for detecting small molecules like hapten. Then it is important to know that what mode of analysis we want to achieve. So ELISA is a tool that can be used for both quantitative and qualitative detection. So we want to determine whether we want to achieve the qualitative or quantitative detection. So based on that, we can choose the ELISA kit of our interest. Next is the sensitivity. So it is very important to determine the sensitivity of the kit. If the researchers have no clue about the concentration of the analyte in their sample, so then in that case, ELISA kit with a broad detection range are a better choice. If the concentration of the analyte in a sample is very low, ELISA kits with very high sensitivity are recommended. And if the concentration of the analyte is too high, then dilutions of the sample may be made to adopt the detection range of the ELISA kit. Next comes the sample size. ELISA kit usually required from 100 microliter to 10 microliter of sample. If the amount of the sample is very low or the sample is very precious, it would be better to choose ELISA kits that require less amount of sample. Next is the recovery and linearity data. So recovery and linearity data assess the performance of ELISA kits. Recovery helps determine whether analyte detection is affected by the difference in sample matrices. The higher the recovery, the better the ELISA kit. The linearity of dilution determines the extent to which the dose response of the analyte is linear in a particular diluent. Ideally, the concentration of the samples should be similar for all dilutions. So most of the suppliers in the market, they provide recovery and linearity data on product specification sheets. Besides these recovery and linearity data, some other important parameters that need to be accessed are sensitivity, dynamic range, and standard curve. So if you can see the product data sheet supplied by the different manufacturers in the market, they mostly provide these parameter data. And it is very important to carefully compare these parameter data, particularly recovery and linearity, to choose the right ELISA kit. Then comes the detection system. There are several different detection systems in ELISA, including colorimetric, fluorometric, and luminescent fluorescent methods. All ELISA involves the immobilization of the analyte to a surface, as well as the use of the enzyme label and the matching substrate. So it is very important to choose an appropriate enzyme and a matching substrate. However, enzyme substrate reaction conditions, the microplate, and the detection device should be properly chosen. So based on the availability of the detection or plate reader in the lab yeah. The next is the ELISA kit should be versatile. So it should be compatible with different types of samples, different types of biological samples, or like humans, serum, plasma, blood, saliva, urine, cell lysates, tissue homogenates. It should have a simple protocol, convenient operations, and short experimental time. So these are some of the tips that are very helpful in choosing the appropriate ELISA kit. So next, I would like to talk about how can we evaluate an ELISA kit. So suppose we have a product description. We have all this data that I have we have all the data that I have just described in the previous slide, that tips for choosing the appropriate ELISA kit. But how can we evaluate if this ELISA kit is appropriate for our samples or not? So here are some of the parameters that needs to be monitored in order to evaluate the ELISA kit. The first is the standard curve. It is very important because standard curve helps us to calculate the actual testing sample value. So it should be linear. So one should really carefully access the standard curve provided with the ELISA kit. Next is the linearity. So linearity is the text index of experimental accuracy and specificity. So to access the linearity of the sample, linearity of the assay, all the samples meaning it can be serum, plasma, and the known concentration of samples were spiked with high concentration of analyte in various matrices and diluted with sample diluent to produce samples with values within the dynamic range of the assay. And the results are, here are the results that are expressed in the percentage observed. So higher the percentage, the better the linearity of the experiment. So moving forward in this series, the other parameters to evaluate the ELISA kits are next is the precision. So precision is the test index of repeatability. So it is the it is measured as the CV percentage, that is the coefficient of variation from the mean value. So the higher the precision, the better the repeatability. And usually two types of precision should be considered: intra-assay precision and inter-assay precision. Intra-assay precision is the reproducibility within the assay, within the wells in an assay between the wells. And inter-assay precision is the reproducibility between assays, which can say batch to batch variation also. Next is next parameter, important parameter to evaluate the ELISA kit is the detection range and sensitivity. So the limit of detection is the test index of sensitivity. The lower the detection limit, the higher the sensitivity. So it is important to select the ELISA kit with broad detection range. Especially if we are unsure about the concentration of our sample, of our analyte in the sample, then it is very important to select the ELISA kit with the broad detection range. Next comes the specificity. Specificity is crucial index to confirm cross-reactivity and interference. So mostly it is important to use the raw materials with high specificity to ensure that the ELISA kits has low cross-reactivity and no interference. And it is a very important parameter, because if our samples, or if the ELISA kit shows cross-reactivity of the analyte with its analog, then our results will not be very accurate. Last in this last parameter to evaluate the ELISA kit is the recovery. So it is the test index of experimental accuracy and specificity. So recovery usually reflects the loss of degree of analyte in the sample in the process. So as you can see here, the higher the recovery rate, the higher the accuracy. And how the recovery rate was determined? So it is determined by spiking the levels of analyte throughout the assay, throughout the range of assay in various matrices. Okay, and then the samples were further diluted and to perform the recovery rate. The recovery rate was analyzed here, as you can see in the percentage. The higher the recovery rate, the higher the accuracy of the kit. So moving forward, I would like to talk about the BioVision ELISA kit portfolio. So BioVision offers more than 500 ELISA kits. So they are mostly available in sandwich or competitive ELISA formats. So, and they are mostly available for, commonly they are available for human, mouse, and rat species. We do offer ELISA kit for other species like goat, rabbit, or they can be, or we do offer kits that can be used for any species that are, or we say like universal species. It can be used for either for mammals or for bacteria or for any other species. We have a wide selection of ELISA kits in various research areas. All of our, I'm going to talk about it in a minute. All of our ELISA kits are available in 96 well format. And they usually comes in a strip format. So we provide 12 strips of 8 well each. So it is very convenient for the users. They can use as many kits or as many wells they want at a time, and they can save or they can store the rest at an appropriate temperature for future use. Mostly our ELISA kits do not require any expensive equipment or instruments. They usually require a plate reader that can read at 450 nanometer. In some cases, we do recommend a reference wavelength of 650 nanometer just to set up the base levels. All of our ELISA kits are very accurate. They shows reproducible results with low intra and inter assay variability. They are they shows high sensitivity and specificity with low background. They all are optimized for broad range of sample types such as serum, plasma, tissue, cell culture, supernatant, urine, and many other biological fluids. ELISA kits are very convenient to use and no special handling or disposable required. All our ELISA kits are thoroughly QC'd to confirm the lot-to-lot consistency or minimal batch-to-batch variation. And we do offer a competitive pricing. Next, I would like to describe describe our BioVision ELISA kit portfolio. So our ELISA kit portfolio are broadly categorized into these categories. So first among them is the cancer research. It is a very important category, you know, like it's a very hot topic for research, which includes angiogenesis, ELISA kits for angiogenesis, ELISA kits for immune checkpoints. It's again a very hot and popular topic of research, and tumor biomarkers. We do offer another category of ELISA kits, which is also very similar and includes which also offers some more product line in this is the traditional ELISA kits for traditional ELISA kits for cytokines and interleukins, immune checkpoints, including ELISA kits for immuno-oncology. We do offer ELISA kits for the quantification of isotypes, immunoglobulin isotypes like IgG, IgGA, IgGM. And we have a product line for the detection of different infectious diseases. We have a very broad product line of metabolism ELISA kit that includes the ELISA kit for the analysis of diabetes related analytes or antigens, drug metabolism, hypothyroidism or obesity, and many more, like including small molecules like CGMP and several MMPs. I would strongly recommend, if you like to have a full list of BioVision ELISA kit, you can go to our website and check the complete list there. Another very popular category that we offer for ELISA kits is a stem cell research that includes another hot topic of biomarkers, ELISA kit for biomarkers. Also, I will describe about it briefly. It also includes the ELISA kits for differentiation and proliferation. Recently, we have added a very interesting category of toxicology, ELISA kits for toxicology, that includes ELISA kit for antibiotics, detection of antibiotics, detection of drug residual, environmental toxins, food toxins, and additives. It's a very interesting category, and it's getting more and more popular nowadays. And another new category that we have recently added is the product as the neuroscience for the detection for the research of neuroscience, for neuroscience research, that includes the ELISA kit for Alzheimer disease, neurotransmitters, and Parkinson disease. So I'm going to talk about them briefly. So first is our category of toxicology or antibiotics. So this is a very popular category, and not many suppliers are there in the market that offer this ELISA kit for antibiotics, to detect antibiotics. It is very popular for the detection of antibiotics, either for quality purposes or for basic research. Some of the popular targets that we offer is gentamicin. It's very popular. Ampicillin and streptomycin. These antibiotic ELISA kits are very popular, and it's getting popular day by day. All these most of these ELISA kits are validated on different sample types. As you can see here, we offer a broad range of we offer a standard curve, and they have a very low sensitivity. And as you can see, that these ELISA kits can be used for different types of biological samples like serum, urine, tissue, plasma. And you can also see that these kits, most of these kits can be used for honey, milk, egg, milk powder. That means that these kits can be also used for food samples, which also makes it interesting for the food industry, for the researchers involved in the food quality involved in the research of food quality, or with the food industry. So next category that we have in the toxicology product line is the ELISA kit for different types of additives. So here we have food additives. Some of the common targets are listed here, and then this is a very wide range of other toxins that we offer, and it's gaining a lot of attention these days. Among this is a very popular is a BSA ELISA kit that can detect the residual BSA in the samples. The other is CHO host cell proteins that can detect the CHO cell proteins in the proteins from the proteins that are expressed in CHO cells, E. coli protein. We do offer the kits, ELISA kits, to detect the drugs like morphines, opiates. So it is also getting very popular as it can detect the drug residual drugs in the sample. Moving forward in this in the ELISA kit portfolio is another category is biomarkers. Biomarkers are generally referred to measurable indicators of some biological state or condition. So as we all know, that they are used to understand the mechanism of action, safety, or toxicity indicators as a diagnostic tool, They can also be used for the selection of lead candidate. So all these assays are very helpful in screening tumor biomarkers or measuring hormone levels in different disease conditions. So BioVision offers a wide selection of ELISA kits, so that you can see here listed here. So major categories of disease categories that we offer ELISA kits are autoimmune diseases, ANA screen and C-reactive protein are a hot topic among these. Then we inflammation, neuronal damage, renal disease, tumor biomarkers, so another very hot topic. Diabetes, thyroid function, oxidative stress, and cardiovascular stress. So you can see that these are the traditional ELISA, these are traditional standard curves for the ELISA kit in this category. This is just few, and if you would like to get the full list, we have a flyer available on our site. Next category in our ELISA kit category in our portfolio is the ELISA kit for infectious diseases. So currently we offer ELISA kits for these targets. This is our current portfolio. So we do offer ELISA kits for different disease targets. You can see here for dengue virus, measles, HSV, mumps. And I would like to specifically mention that we have ELISA kits that can detect the different immunoglobulins in the samples. Like we have a ELISA kit for IgG, IgGM, IgGA for different diseases. So these can detect IgG, IgGM, or other immunoglobulins. Yeah, so these ELISA kits are very sensitive and are very specific. So there is no cross-reactivity from one virus to another in these that is shown in the essay. Next category in our portfolio is another very new category that we have recently added is the ELISA kits for protein tag proteins. So currently we offer ELISA kit for three different tag proteins: flat tag protein, GST tag, and His tag. So as you can see that this is the detection range. It offers a quite wide detection range, and it can be used for different types of samples, E. coli or mammalian cell lysates. His tag protein ELISA kit can be used for bacterial, insect, and mammalian cell lysates. So this is also one of the product line that is becoming popular day by day. It is very useful if you want to detect the tag protein in your sample, the residual tag protein in the sample. So it is very helpful for the life science researchers that are working with the recombinant proteins that are expressed in different systems like E. coli or that are expressed using these different tags. Okay, so next, one of the most popular category in our portfolio, ELISA kit category in our portfolio is the ELISA kit for biosimilar antibodies. These antibodies are very helpful for the researchers who are working on the development of biosimilar drug, or who are studying the effect of the biosimilar drug in different diseases. So this is the common principle of this ELISA kit. It is based on sandwich ELISA. As you can see here, So the antibody against the biosimilar drug is immobilized into the microvell plate. The biosimilar antibody is added into the well, and then which was detected using a secondary, that is anti-human IgG Fc conjugated to biotin to detect the signal. It is also very important. It is also very helpful for the researchers who are studying the dose-dependent pharmacokinetics of these biosimilar antibodies. These ELISA kits are very helpful to identify the biomarkers for risk factors of these antibodies, and also the adverse drug reactions. They can be used for different cell types like serum and plasma. So this is the typical standard curve for biosimilar antibodies. Yeah, so as I mentioned, these are very helpful for the life science researchers who are working either on drug development, or the basic researchers, or they are trying to study the effect of these biosimilar antibodies. Another related category with the biosimilar antibodies ELISA kits is our biosim ELISA kit for anti-drug for anti-drug antibody. So this is the product line that can detect the antibody against the biosimilar drug. So this measures the anti-drug antibody. That means the antibody against the biosimilar antibody or biosimilar drug. This is also very helpful for the researchers who are studying the immunogenicity. For example, they are trying to understand the immune response to the biosimilar drug. So this is again the very similar principle. So the biosimilar drug is coated into the microELISA plate. Then anti-drug antibody is added, the sample containing the anti-drug antibody added to the well, which binds to the biosimilar antibody, which can be further detected using secondary antibody, that is anti-human IgG FCC conjugated to biotin. I would like to mention this principle because this is one of the most frequently questions that we get from the researchers, that what is the principle of this kit and what it can detect. So it's kind of a little confusing. So that's why I would like to specifically mention that these two kits, we have two different product lines. The product line, the ELISA kit first, that can detect the biosimilar, and we have the ELISA kit that can detect the antibody against biosimilar, the biosimilar and antibody against that biosimilar drug. So this is also, these ELISA kits are very helpful for the researchers who are trying to understand the immune response against these biosimilar drugs. They can be also used for different types of samples, but most commonly are serum and plasma. We have quite huge portfolio of these biosimilar drugs or anti-biosimilar drugs. Again, I would strongly recommend go to our website for the complete product list. Next is we have product line for ELISA kits for immunoglobulins quantification. So currently we have ELISA kit that can detect IgG-A, IgG, IgM, IgG, IgGM in different species: human, mouse, rat, rabbit, and porcine. So we are currently adding more kits to this product line. And if you have any customer who would like to have any ELISA kit for any other immunoglobulins for any other species, please feel free to contact us. Maybe we can help you in providing that. Next is the category of ELISA kits that I would like to talk about is another very hot topic of research these days is immune checkpoint or immunotherapy using immune checkpoints. As you know that the PD-1 discovery of PD-1 and PD-L1 after the breakthrough in the field of immunotherapy. Currently, we have added a few more targets that are again getting more popular, like CD20, CD27, 152, CD223. These are all newly added, and we are still working on this product line and adding more and more targets as it is available for research. Yeah, so this is very useful for the researchers who are trying to who are doing research targeting immune checkpoints or immunotherapy. Next is a fairly new product category that we have recently added is ELISA kits for neurotransmitters. These are the ELISA kits that can be helpful in detecting the neural response that has been regulated by these different analytes. So some of the common targets in this category are adrenaline, serotonin, dopamine, acetylcholine. These are some of the histamine, very popular targets. And this is also very few. They are available for different species. Some of them are available for universal that can be used for any species, and it also offers a very broad detection range. Goes up to the picograms. You can see this is the flyer for the neurotransmitter kits that we offer, that's available from BioVision, and you can find this on our website. So at last, I would like to talk about our product line of ELISA kits. This is called Quick Detect ELISA kits that is highly optimized to offer a short overall short experiment time or less experiment time. So these ELISA kits, these kits can be completed in less than 90 minutes, and they get quick results and very price competitive. These kits are very time saving. They are mainly based on a sandwich ELISA format and offer high sensitivity and specificity. So these are some of the popular targets that are available in this Quick Detect ELISA kit format. These kits are highly optimized. The capture, the detection process, the incubation time, the washing steps, the blocking steps, everything is highly optimized to either to shorten the time, the incubation time, or to offer this simultaneous or these are optimized that several steps can happen simultaneously, so that the entire ELISA kit can be completed in a shorter time, in just 90 minutes. So currently we offer few of the popular targets that we offer in this format are listed here, and for complete list I would again recommend you to go to our website, and you can find the complete list there. So now I would like to sum up this webinar by giving you some of the experimental tips. So if your customer has some problems using our kit, or you know, they are or they were trying to setting up their own experiments and they are not getting the desired results. And I know that it is often very frustrating and it's very time consuming. So I would like to share some tips that can be very useful uh, to use these kits to achieve the desired results, or to, you know, to get the maximum out of these kits. So first of all, it is important to find the right product. So for that, it is important to know your target, whether it's you know, like or and to find the kit that is compatible with your target. So that if you're using a sample type which is a serum, plasma, cell culture, supernatant, or tissue homogenate, so it is important to determine the kit that is compatible with your sample type, because not all kits are compatible for each sample type. And then using these kits can be just a waste of sample time, and the time and consume, and it requires a lot of energy, which is like just waste of energy. So it is very important to know your sample type, your target, it is and as not only sample types, it is also very important to know the species, that whether your kit can detect your the species of your sample. Because we often get these questions that, hey, I can see that you have MMP3 kit. Do your kit can detect rat samples? You know, but if the kit is for human samples, how can the kit can detect rat samples? So it is very important to determine to find the right kit that can detect the right target type, the right sample type, and the right species, as well as the detection range. I would strongly recommend that look for the kit that has the broad detection range if you are not sure about your sample, because that can be, by that way, it can be it is always safe to have the broad the ELISA kit with broad detection range, you know, just to make sure that the samples can be um, can the samples can be detected within the range of the standard curve. Then it is, after that, it is always recommended to run a pilot experiment and standard curve. So along with this, it is always very important to determine the detection range of the kit. As I just mentioned, that the broad detection range can offer, if ELISA kit can offer a broad detection range, it is always good to go for it. If the customers are unsure about the concentration of the target in their samples, it is always good to select the kits with low sensitivity, high specificity, with low cross reactivity, and here are some of the this is not the accurate dilution or concentration, but just the broad overview. So if the samples are highly concentrated, then it needs to be further diluted, just to make sure that the concentration of the sample is in the range of the standard curve. And it is also very important to include the dilution factor in the calculation. And furthermore, it is also very important to use how you prepare the sample and how you store the kit. So it's very important to run the assays as soon as possible after a sample collection. Avoid freeze thaw cycles, run duplicate for all samples if possible. We also recommend to run standard curve each time. It's not that standard curve should be run only once. Please run standard curve each time you perform the experiment. Yeah, so these are some of the experimental tips that definitely help to improve the results and the performance of the kit. With that, I would like to conclude this webinar. Um, so, um, come, if you would like some of the um, literature of this product line is available on our website. Please go to our website for the flyers and brochures if you are interested, and feel free to share with your customers. If you want to have the hard copy, please feel free to reach out to me or to our marketing team, and we will definitely send you the hard copies. So these are some of the flyers that are listed here, but if you go to our website, you will see the complete list of all the ELISA kits in different research areas that we offer. And with that, I would like to thank you all for attending this webinar, and I am happy to take any questions. So I'm happy to take any questions. Please feel free to type in the Q&A box. Yeah, so okay, so we got this one question from one of our attendees, and they would like to know that how the BioVision kits offers flexibility and versatility of products. So actually, by versatile, I would say that the kit can be used. Actually, it's not, these both terms are go hand in hand, like it's go with each other. The Okay, so yeah, so the question is how our kits determines the flexibility and versatility of the products from BioVision. Yeah, so versatile means that the kit can be used for different types of sample types. That the kit can be used for um, for for serum samples, for plasma, for cell lysates, tissue homogenates, urine, saliva, different biological samples. That is called that the kit is versatile. So that's what this question we often get from our customers, that whether this kit can be used for this kind of samples. They have very strange type of samples. So the kit, our kits are highly optimized, that can be used for different sample types. So I hope this answers your question. Is there is any other question? Uh, yeah, so yeah, definitely. This PowerPoint slides are available. So we will send you via email. So these PowerPoint slides and the presentation and the complete recorded webinar will be sent to you via email. Just send us an email if you would like to receive it. Yeah, you can reach out to me on my email address, or you can reach out to tech@biovision.com. We will send you the recorded webinar and the PowerPoint slides. Okay, definitely. Thank you for sharing your email address, and we will send you the PowerPoint slides. So, well, if there are not any more questions, then I would like to end this session. And thank you again, everybody, for attending. Thank you so much.